A very good evening, friends. I'm Dr. Saurav Arora, and I'm the founder of Initiative to Promote Research in Homeopathy, which is an online portal for promotion of research. And today we are going to discuss about how to write a clinical trial in homeopathy. Now, this has been a very preferred and very apt topic to take at this point of time when homeopathy is struggling with plausibility from clinical point of view, from fundamental point of view. And many a times we want to write something, we want to do something, but we have some hitch or we have some obstacle. So today we are going to discuss the same. Now, the by end of this webinar, I would try to provide a brief about clinical trial writing, its areas, its strengths, its weaknesses, a brief about research methods, limitations. But because since we are limited with time, so this webinar does not cover the design part in depth. And after this webinar, the live chat, which is open for half an hour, would be available at my website, that is researchinhomeopathy.org. Before proceeding further, there are some sections which I found apt to discuss at this point of time, and we would be discussing those before initiating a formal discussion of clinical trial writing. When we talk of research education, we have three aspects: the research literacy, the research proficiency, and the expertise. And according to these level of literacy or the education, one has the decision to undertake a certain level of evidence. Maybe if we are very proficient, we can take case control studies or cohort studies or RCTs. If we are expert, we can take systematic reviews. So this thing is imbibed in each and every one of us. We are either proficient, we are either expert, or we are learners. And accordingly, we should undertake the research. Why clinical trials? This is a very good question. Why trials? Before going why, let me tell you that clinical practice is different from clinical research. And many a times we want to combine both the things. We want to combine clinical practice with the clinical research. But there are many differences. But the major difference which I found that in clinical practice we use established treatments, while in clinical research we collect evidence to establish treatment. Now there's a difference. For example, if I say that for headache I'm giving belladonna. Now in such case belladonna is an established therapy. It's an established treatment. I'm giving it for headache. But if I flip my question that I want to see the role of belladonna in headaches, now the question comes. There, I need to collect evidence to prove this statement, to establish this treatment. So, in clinical practice, in clinical research, there's a flip. Clinical trials, if we talk of formally, that they are the research studies that explore whether a medical strategy, treatment, or device is safe and effective, obviously for humans. And these studies may also show which medical approach work best for certain illness or a group of people. A classical definition says that it is divided into four phases. That is 1, 2, 3, and 4. Like this. Effect on body, that is preclinical or phase 0. In human beings, if we talk safety, that is phase 1. Effectiveness at treating diseases, phase 2. Larger studies, phase 3. And long-term study, phase 4. So this is in a nutshell about the phases of the study. Now, coming to homeopathy, they are the preferred study design that warrant to prove the evidence-based nature with a rigor. Rigor is required, and these are needed to prove the biological effects of homeopathy in human beings. RCTs are considered as gold standard, but let me tell you, they are not the only one. They are one of the study design which are gold standard, but there are other study designs which we must adopt and learn to use in homeopathy, because research in homeopathy is not up to that mark. Now, is trial writing a very difficult task, or is it easy? We need to identify the problem statement before we try to write the clinical trial. Say this is the first step. We want to write a clinical trial, but we should know on which topic, on which area we want to venture. That is called the research question. It is the first step of that research. That comes from area of choice, area of interest, observed phenomena, or area of uncertainty. For example, if I'm doing PhD, I have a choice that I can work on homeopathy and establish its treatment modalities. Being a clinician, being a simple plain clinician, not well versed with the research this thing, but I have interest in research. At some places I have observed certain things that yes, this might help in such cases, or this medicine might help in tumor cases or cancer cases or headaches and migraines, whatever. That is observed phenomena, but I want to test that phenomena to prove the efficacy of homeopathy, or any area which we are uncertain. There are many places where people claim that this medicine gives wonder, do miracles, but we are not certain. We must take a clinical trial in that area. That is the research question, where it comes from. A research question, a good research question should meet the final criteria. And when it meets the final criteria, it becomes a good research question. That is feasible, interesting, novel, ethical, and relevant. Can it be investigated without an undue amount of time, energy, or money? For example, if I say I shall explore the depth of sea to see the Titanic, no, it is not possible. It is not that easy. It is not that feasible. But if I say I shall observe the movement of whales during daytime, yes, that is quite feasible in an area where I have the access. It should be interesting and clear. For example, if I say the role of homeopathy in improving pulmonary function test in emphysema, yes, it is interesting, it is clear. But if I say role of homeopathy in diabetes mellitus, see, diabetes mellitus, obesity, PCOD are very vast topics. They cannot be covered in nutshell in a shorter span of time. So we must have clarity what aspect of that disease we are studying, because these are syndromes. It should be novel. Is it worth to undertake in terms of time needed, energy required, effect on or for participants? Novelty is the prime thing. And the question is all about ethics. It will not involve physical or psychological harm or damage to human beings or to the natural social environment of which they are part. We are not here to trouble people. We are not here to play with the ethical issues. We are here to help, being homeopaths, being practitioners of the healing art. How relevant is the question to the science, practice, and society? It can only be achieved if we know the previous work which has been done in the same area. Now our research questions become the finer, the good research question. It is feasible, it is interesting, it is novel, it is ethical, it is relevant. Once we have identified that research question, we have tested that as per the final criteria, we must make a hypothesis. Hypothesis is a formal affirmative statement predicting a single research outcome. Many times you might have seen the multiple research outcome hypothesis. They are the complex study design, but usually it is a single research outcome affirmative statement. It's a tentative explanation of the relationship between two or more variables. It is directional. Now, I think this is getting a little complex. Let us take examples of hypothesis. Homeopathy enhances the effects of scaling and route planning in patient with chronic periodontitis. Now, this is feasible, this is interesting, this is novel, this is ethical, this is relevant, and this is a hypothesis. Water has memory. Homeopathy prevents mastitis in organically farmed dairy cows. In all these examples, we are taking the relationship between the variables and they are directional. Now, the research accept or reject the hypothesis. Many times you must have seen we have null hypothesis and alternate hypothesis. Now, null hypothesis says that homeopathy is not effective. Alternate says it is effective. So the research reject that null hypothesis, means it reject that says homeopathy doesn't work. we have to do in such a way that it accepts or rejects. This aspect is also important, the literature review. Why I'm talking of this literature review, I will try to correlate all of my discussion in the later part of the presentation. A literature review is a discussion of your knowledge, the topic under study, which is supported by the research literature. This is a foundation. Until unless we don't have a very good research question and a foundation, we cannot undertake a clinical trial. Even if we take, that is bound to fail. It is not, which we usually do, it is not a study by study or article by article description, or it is not a restatement. It is also not a brief review of articles. We are collecting 10 articles at one place, presenting them in the form of literature review. No, it is not that. What is it? It is a broad, it is comprehensive, in depth, systematic, critical review. Broad, comprehensive, in depth, systematic, critical, scholarly publications primarily, which have been published in peer reviewed journals. You may cite unpublished scholarly print materials, audiovisual materials, or personal communication if you don't have those scholarly publications. This reminds me of an example that I submitted one paper, one case report on endometrial polyp, which was published in American Journal in 2012. At that time, I could not find a single reference of use of homeopathy in endometrial polyp. So I had to take citation from the other sources from conventional research about endometrial polyp. Moving further, this review of literature has implications on every aspect of education, research, practice, and theory. This is basically a amalgamation of all these in a concise manner. You are picking observation literature from each of the aspect, and you're jotting down in one place which is logical, which is the foundation of your study. Now, it determines what is known. Is there any duplicity? Is there any gap? Is there any consistency or inconsistencies? Strength or the weakness? Or research design or method? You know that my literature reviews says that up to single blind study has been taken in this area. Now my aim would be to undertake the double blind study, not to waste my energy in single blind of the observational studies, because much of the research work has been done in that area alone. Therefore, I have good foundation. I know that these designs have been tried in the past, and my research is ahead of them. So it provides a motivation for future work. And there's no point spending time and energy for work which is already being done. But yes, in some cases we may retest that thing also. Some cases, if we have a challenge that this thing is perfect, we can retest that, but that's a separate scenario. In research, you always aim for the higher designs. These are some of the general guidelines I would like to discuss with you, that you should sit with an aim to point out a major research topic rather than a global problem. For example, number of home pads in all countries versus number of home pads in India. Obviously, I'm an Indian. I can I can still try for number of home pads in India, searching for state to state, but I cannot predict the number of home pads in all the countries. This is not my aim. My aim should be very narrowed down. Like I said, it should be very concise, clear. Discuss this topic with your peers, colleagues, and mentors. Once you have a research question and a foundation of your study, and you are about to start a clinical trial or you're about to write a clinical trial, you must discuss with your peers. For example, MD batch students, they must go to their guides and discuss the things. But don't attempt to cover each and everything. Pick up the most relevant research. Sometimes we try to take this also, that also. That is waste of time. We should be very focused, narrowed down to our research question and foundation of the study. Broadly, the research methodology goes like this: that is, specific objectives, preparation of protocol, the data collection, analysis, interpretation, and publication. Today, we are limiting ourselves to preparation of the protocol. Now, the process. Now, from here henceforth, we will discuss the clinical trial protocol preparation process. And for the people who have just joined in between, the webinar is, I'm just giving in brief that this is to provide a brief about clinical trial writing and about research methods. And after this webinar, the live chat is open for half an hour at my website, that is researchinhomeopathy.org. You can go to that, you can ask me any questions. Now, we are moving to section wise discussion. Why section wise discussion? Because writing should be methodical. Let us discuss each and every section, how these sections are formulated. First and the foremost is introduction. Introduction should be simple yet informative. Don't make a complex introduction. Many times I have seen people write complex statements, complex starting points of introduction. No, it should be as simple as it can be, but informative. It must clearly state and illustrate the background work done. Now, this background work done is obviously will come from the literature review. It must include a summary of a search, that is literature review, to indicate why this proposal is necessary and what it aims to contribute to the field. Now, you can correlate. If we have done a good, thorough literature review, we can clearly state the background, we can clearly state the summary, and that is actually important for the proposal. This section should end with a brief statement of what is being proposed to be done in this research. I'll take an example. It's a funnel shape criteria. You start with a broader thing, that is background information. Whatever the study have been done, the preclinical studies, the prior research work done in this area, the rationale or the need of undertaking a study, and proposed or the expected role of homeopathy in such condition. So this is a funnel. From broad, you are narrowing down to this thing. For example, obesity is one of the global problem nowadays. XYZ research has been undertaken in obesity, but homeopathy has been found to be effective in obesity in people having a certain background. And this study aims to see the role of homeopathy in this age group of obese people with this aim. So this can be, a shorter, crisp introduction. After introduction, you must define the study objectives. Objectives are primary and secondary. These are very sensitive things. Many times we write many objectives: one, two, three, four, five, six objective, maybe seven objective, A, B, C, D, E, F. But there should be only one primary objective, and that should be the overall purpose of the study. It should be clearly and concisely defined. It should be related to the research question, to see the efficacy of homeopathy on TSH changes in hypothyroid subjects. That is the primary this objective. But there are certain other things in addition to main objective which we like to study. For example, in diabetic patients, along with blood sugar level, we want to study lipid profile changes, which was not our research question, but we want to see these changes. These become the secondary objectives. So always in your research, identify a primary objective and identify secondary objectives, because the data analysis will be primarily based on the primary objective. If the primary objective is very clear, very concise, very one to one, the statistical analysis would be easy and impactful. But if we take that to study the role of homeopathy on TSH changes, to study the role of homeopathy on quality of life, to study the role of changes in weight, all these things are separate entities which we have to analyze separately in statistics. So this becomes difficult. So always the primary and the secondary. Study design. Although we are not going into depth of this thing, but let us have a quick word about study design. Basically, it can be experimental or observational, that where you intervene and where you don't intervene. And usually, like we discussed, that it can be 1, 2, 3, and 4 phases in conventional trial. But in homeopathy, as we don't have any new molecule identification or toxic or safety study, we don't have stage 1, stage 2 clinical trials. We straight away start from the trial 3 and phase 3 and phase 4. This is a pictorial depiction that it may be observational or experimental. An observational, we may have a comparison group or no comparison group, that is descriptive study. Or if we have one comparison group, that is case control or cohort study. While we have experimental design, we can randomly allocate. If yes, this is the randomized controlled trial, and it can be non-randomized controlled trial. Once we are done with the introduction, the objectives, the third most important thing is outcome. A measure of the quality of medical care, the standard against which the end result of the intervention is assessed. What we want to measure, what we want to see, observe in our trial. To undertake research effectively, always choose objective, that is, you know, the outcome which can be objectively categorized, such as values, for example, TSH, hemoglobin, standardized parameters, and such, because these things are can be easily standardized, observed, recorded, evaluated. But if I say, okay, fine, the patient says my headache is better, better is not an outcome. You cannot statistically assess better until unless it is a validated score. It is a part of that score system. There may be subjective measures which can be standardized and measured, which you can take, for example, validated scores, quality of life. In those cases also, the symptoms or the signs are converted to numerics, because numerics can be easily assessed, recorded, and analyzed. So the better the outcome you have, the better the research you have, and they should be validated. You must not create a new outcome measure. You can create, but the design would be different, the approach would be different. If you're doing a clinical trial, established clinical trial, always try to have an objective which is well validated. No, they this we have to write in our trial about the sensitivity or specificity of these outcome measures. More reliable the investigation, more stronger is the evidence. We know that these have these specificity 99%, they have sensitivity 96%. We can assess them easily. Also, when we are writing the outcome measures, we should write the repetition of investigation. How frequently we shall undertake the investigation, and chart of such follow-up is to be presented in a tabular or the flowchart form for easy understanding and easy comprehension. For example, I'll give the example later. Endpoint. Now, after objective outcome, we have endpoint. Endpoint is a point marking the completion of a process. In research, we have primary and secondary endpoints where you will stop your research. Primary endpoint, when the process is completed or we have achieved. For example, TSH came to normalcy in hypothyroid patients on or before the completion of study. Our target was that when the TSH would come down below 3, our primary endpoint should be achieved, and it came below 3. So our primary endpoint has been achieved. But there are certain scenarios where we have to take the secondary endpoints, like completion of the study time. We followed patient for 1 year, but TSH didn't came to normal, or it came to normal. We kept on recording those values for 1 year. So our second primary endpoint was to see the value changes, and the secondary endpoint was to then the study times complete, or when the participants were withdrawn from the study due to any reason. We will discuss this aspect also, when to withdraw a participant from the study or any adverse event has happened. So those are the secondary endpoints. Okay, now a trial should have a single endpoint based on the one outcome measure. This is why we told that outcome measure should be validated with a valid endpoint. Now, endpoint can also be the time taken for an event to occur. For example, many times many researchers ask how much time homeopathy takes to taper down the changes in TSH or sugar, or how much time is needed to increase the hemoglobin level. You say that it may be 1 month, it may be 6 months, it may be 1 year. But when in research, we say that in our patient, we have seen that within 3 to 6 months, within 3 months, within 6 months, within n number of time, the changes started occurring. So that endpoint was taken as time to event value. Proceeding to population, we have defined outcome measure, we have defined endpoint. Now we must decide the population. What should be the inclusion criteria, what should be the exclusion criteria, what we are going to consider, what we are not going to consider. And these are fixed, these cannot be changed throughout the research. Other things can be taken care of, but these inclusion and exclusion must be fixed. For example, in diabetic patient, if we say that we will take young adults, both genders, not suffering from any other medical problem. So these are the inclusion and the exclusions. Once we have decided with the inclusion exclusion, we should always propose a sample size. There are different ways to calculate a sample size, and it must be calculated. It is very arbitrary to say that we will take research on 30 people or 50 people or whosoever comes to our research. That is of no value. We must be determined with the sample size, and we can easily calculate. There is one website that is r-project.org, that is a statistical software R's website, where you can go and download the free software, which is a sample size calculator and a number of things you can do in that. And if the sample size is calculated on the basis of previous pilot or clinical research, you must give a clear explanation why it is there. Moving ahead, there comes the screening or the recruitment. You have identified the outcome measure, the endpoint, the population. Now, how will you recruit these population into your study? How will you screen them? Because you cannot invite anyone to come to your study and you will recruit them. You must have that filter, that screening process for the recruitment. What would be the source of patient population? Would it be OPD or IPD or hospital or peripheral OPD or home, whatever? That source must be defined in your trial. The screening process must be defined. How would you be able to filter them for the recruitment in the study? For example, camps. We do camps. We went to an area, we organized one camp, and under that camp, we convince people to take part into our research as per their inclusion criteria, or we excluded them to come to our center because they were not fit. And when and how we would be recruiting the patients in our study, there may be a time gap between screening and the actual start of study. There are times when we say that we have screened the patients, we have counseled them, and within 3 months we are going to start the study. Or we can say that we have screened the patient, and as soon as the screening is over, we are recruiting them within trial. Where the treatment and follow-up shall take place, the study site. It must be a registered and well-equipped center as per your research question. You are undertaking a trial on neurology and you don't have MRI access in your center or outsource, that is of no value. If you have an inbuilt lab, that is the most good thing. But if you don't have, you can outsource. But that study site must be appended, fixed with all the necessary things to undertake a research. We have identified population. Now we want to allocate them randomly. This is the part of the trial which is most important part. It is a method of assigning participant to either treatment or the control group. It is done to remove the bias, because the patient who can recover, put them into medicine group. The patient who critical patient, place them into placebo group or the control group. This is a bias. So we do randomization to remove that bias, subjective bias. Allocation bias, it may be easily done using a software available online, or you can use your desktop, or you can flip a coin. There are many methods of randomization, and this is one of the website I found that says saghaei.net, where you can find Find a open source random allocation software which you can freely use in your clinical trials. One thing, control group is not always a placebo group. We have a myth that if it is a control group, it is a placebo group or nocebo group, but it is not. Control can be a standard conventional treatment against which we want to see the efficacy. For example, we say we want to see the efficacy of homeopathy in comparison to standard painkillers. The standard painkillers becomes the control group. You must describe your intervention and control groups thoroughly and their allocation, because this is the foundation which would be used for statistical analysis along with other things. And usually it is preceded by the blinding if required. Regarding blinding, I would like to suggest that always do blinding in presence of a third party or a person who is not involved in your research, because blinding sometimes may mimic subjective bias. We may disclose that information to a statistician, but blinding is not done in the presence of statistician. Not, don't do this. Now, as per Consort guidelines, which is the consolidated standard of reporting trials, which is available at consort.statement.org, we must put a flow diagram of enrollment, screening, and allocation. This we have discussed in words. If we have this in flowchart form, that becomes easier to comprehend. For example, this Consort guideline chart, that enrollment phase, allocation phase, follow-up phase, and analysis phase. In enrollment, we discussed the what is the eligibility of participants who were excluded. Now they were randomized and put into two groups, that is intervention group and the control group. And details of those allocation groups. How would they be followed? And what would be the analysis criteria? So this is a consort flow diagram which must be inserted in a clinical trial protocol. After all this, we have four important things, that is PICO: population, intervention, comparison, or the outcome. These four combination makes the most important parts of our research. Whom would we be studied, with what intervention or exposure, in comparison to whom, and on what basis? We would be comparing two group of people, one on allopathy, one on homeopathy. We would give one group homeopathic medicines and we would compare that with the conventional group on the basis of changes in glycated hemoglobin. So this is PICO criteria. One was final criteria we discussed earlier, and this is the PICO criteria, PICO. Moving to follow-up, it is the most important aspect. Why? Because a well-planned research can only be executed if we have a well-planned follow-up, and we must choose the follow-up frequency, which is based on our research question. The follow-up may be depicted in tabular or the flowchart form, which is easier to comprehend. Also, like we inserted consort guidelines, we should insert the follow-up frequency table, like this one. On the left hand side, we have all the variables. On right hand side, we have monthly follow-up, and we can tick what is to be seen every month, every 3 months, every 6 months. Basically, a follow-up frequency. What all? For example, in this, the TSH would be studied at baseline at the end of 3 months, 6 months, 9 months, and 12 months. But the blood pressure would be recorded on each visit. After this, we have treatment part. This part of protocol writing is important from homeopathic point of view. What are the study drugs? Whether the study intend to use a single or the multiple drugs. What is the regimen? How and when the patient would be treated? From where the medicine would be procured, the source of medicine, and how would they be administered? For example, oral or olfaction. On what basis of they would be given to the patient? Is it repertorization, keynotes, specifics, family history, whatever? How we will select the potency dose and how we would change the prescription. These are all things we must tell in detail in our clinical trial protocol, because that is what is making that trial different from conventional trial, the homeopathic part, that we will study this drug with this regimen. We will procure from a GMP certified company or a registered company. We will give through oral route. The selection would be purely on repertorization basis or keynote prescription basis. The potency would be selected on homeopathic principles, and the dose would also be selected on the principles. And we will change this prescription as per need of the hour, as per these criteria. So these criteria must be defined in your protocol. We also mention the steps to maximize adherence and retention in a clinical trial, because it is very common for participant to withdraw from a trial, and that is dropout, which is a valid and practical phenomena. You cannot uh, you cannot request all patients stick to you. They might have some problem, maybe they are shifting to some other places, they may be disinterested, they may have some family problem, they may have lack of time, etc. They will drop out. So what would you do to increase this compliance and monitoring? A good clinical trial always tries to minimize the dropouts by taking quick decisions, adhering to the protocol, maintaining a good rapport with the participant. This good rapport is most important. If you don't have a good rapport with the patient is not going to stick to you. He's liable to go away from the trial. Now, simple method in this section of the clinical trial protocol is to send a reminder to the participants via email or phone. This is the most common thing we do in a trial, that we always tell them through email or the phone that your medicinal follow-up is due, your investigations are due, you should report on time. But writing letter may also be adopted if patient is shifted to some other place or if it's not reporting despite emails or phone calls. Many a times a patient is not clear about research, his and then he's liable to be dropout. He is not under, maybe he's convinced in the first visit that yes, you are doing something with homeopathy and you will treat him, you will make him all fine, but he's not clear about what you're doing. And by the second or the third visit, he does not develop rapport with you, therefore he dropped out. So always initiate a healthy discussion with the patient, because patient usually hesitate to discuss these issues in few initial meetings. You must clarify them that we are doing this thing for your benefit, for system benefit, for homeopathy's benefit, and this is how we are going to go. If you have any query, you can call us anytime, you can ask us anytime. That is how you actually improve the compliance and the monitoring. Withdrawal is also a very common phenomena. How and when the patient would be withdrawn? Will it be early withdrawal? And what would happen to the data collection when the follow-up have withdrawn or the participants are not interested? The withdrawal can be from both the sides. Being a study coordinator, I can withdraw a patient from a study if I think that there is some adverse event or there's the patient is not fit to join that. Patient may also withdraw from the study because of n number of reasons: dropouts, shifting, health issues, financial issue, and number of things. But we should always collect data of such withdrawals and follow-ups, because for example, imputation method in statistics for missing data may be used for analysis. So if we think that this data is useless for us, it is not so. Even that single follow-up can be useful data for statistics, but that should be clearly reported and clarified. Statistical plan must be written in consultation with the biostatistician, because he is the person who will help you for the sample size determination, how you would arrive at a sample size, what statistical methods would be adopted according to the design, because you have given him a research question and an outcome. He will suggest that these results, the expected results, may be determined and processed according to this method, which becomes easier for studies in the longer run. What population is used for analysis? Because there will be dropouts or withdrawals. What kind of statistical analysis shall be done in such cases? Because in research, each and every patient is important. from data point of view, from ethic point of view. Coming to safety and adverse event topic section. After monitoring, we are moving to safety and adverse events. See, all these topics, the sections must be clearly written in your trial, clinical trial protocol. This is a part of protocol. Definition, you must write the definition. What is an adverse event in your trial? For example, in a trial where people are exposed to certain risk, developing a disease is an adverse event. But in homeopathy, we don't see such adverse event in our trial. But yes, we may define that these kind of things would be taken as adverse event in our trial. It may be related to our trial or maybe related to participants also. For example, any things related to mental, physical, psychological, or financial sphere. Patient started following to us, and over the time he thought that he is taking too much leaves from the office and he cannot do that. He withdraw from the study. He is paying. He is being deducted from his salary the days he is coming to our trial for follow up. That is that may be an adverse event, or maybe some patient develop rashes or some skin diseases or anything that is not part of the disease. The reporting must be clear and in simple language. Don't make it a complex. Just write simple that this event occurred, and let the team decide that whether it's an adverse event or an unanticipated problem. Like we said, that blinding. In this case, unblinding procedures must be followed and studied, because in case of adverse event or unanticipated problems, we might have to unblind that patient from the study to know whether it is because of any medicine or because of the placebo. Because in case it is a placebo, it doesn't have to do anything with the medicine or the intervention part. So that procedure blending must be written clearly in protocol before the initiation, and it must be kept in safe with an independent party or institution's head who is not a part of study, to remove the bias. A principal investigator, a course coordinator should not keep that unblinding documents with him. As an ethic, they must be kept somewhere else, in somewhere else custody. It is usually done in the presence of third independent party, maybe a clinical trial agency which you have hired, or bench of expert. You can make an expert team and present to them this unblinding procedure, but involvement of statistician must be limited. He may be made aware about the blinding once the statistical results have been processed. So at this stage, there is no role of statistician. Statistician's role is in designing part, not the unblinding part. And the stopping rules must be defined when you will stop the medicine. This is also an important and very sensitive part, that is data handling and record keeping. What measures you would take to keep it confidential? How would you process this source document to keep them safe? For how many years you will retain the records? And how would you do the study monitoring, auditing, and inspecting? Basically, some of these things are done by the third parties, that are the clinical trial agencies. They have bench of experts, they have tools, they have techniques to keep the identity, the data confidential. The source document, they are retained for certain couple of years, and they do monitoring, auditing, and inspecting, and they provide reports to ethical committees and your team. But this can be done with by your team also. You can hire, you can invite experts to do the study monitoring, auditing, and inspecting, and in turn they will they will give you the report. That is a part of monitoring, but that must be clearly defined in the protocol, how and when this study monitoring and data handling would be done, at what point of time. A study, as we said, it is an ethical consideration. It must be approved, and this approval must be taken by an ethical committee, which is a mandate nowadays. And preferably, your study must have a data and safety monitoring board, that is DSMB. Your trial must be registered with a clinical trial registry, and almost every country has a clinical trial registry. Now, as a mandate, the clinical trials, they must be in that registry as soon they are initiated. The process is simple and clear. You can just go to your Google and search for your country's ethical board, and those clinical trial must be ethically approved and uploaded on the clinical trial registry. This ethical approval may also be taken from some other institutions ethical board, which is a verified and well established board. Coming to study finance, if we talk about study finance, which is an important part, without any finance, no study can take place. It must clearly define the logistics and the manpower, the budget requirement, which should be headwise and item wise, with a detailed breakup year wise, and the total grant in it required for the study. For example, if we see this table, we have manpower, equipment, study material, non-recurring expenditure, or the recurring expenditure, travel allowance, or any expense. And on the right hand side, we have first year expenses, second year expenses, and the total expenses. These study finance, if you clearly define them as per headwise, it would be easier to track them, make an expense statement, and ask for the grants. After all this, in a clinical trial, the important thing is publication plan, where you intend to publish the study results. That must be defined in a peer-reviewed journal, in a magazine, in such set of journals, on complementary journals, alternative journals, and by what time would you be processing data and do the publication. That is also indicated in a clinical trial, because a clinical trial is not an ongoing, endless process that must end somewhere. So usually people take 6 months time for things to get published in a peer-reviewed journal, which is a usual and common time. Always define the timelines or the milestones of your study. This. For example, the screening would take place for 3 months. The data collection would take place for 12 months by the end of this month, by the end of that month, and after that we would take 3 months to analyze it statistically, and we need 6 months to get it published. So all these milestones are to be achieved on time, and if you start well on time, once you have done the recruitment thing, that is over, which is the most difficult part. Usually people attain other milestones on time. We have created a protocol, clinical trial protocol. Now the amendment, it should not be amended, first of all. But in certain cases where it needs to be changed, it must be done by constituting a board, which is which should consist of all stakeholders and legal consultants. And this amended protocol must be reapproved from ethical committee and resubmitted for clinical trial registry. Many a time we make a mistake. We amend the protocol without approval and we don't submit to ethical committees. In such cases, you cannot go for peer review publications or trial completions. Many a trials they fail because of these things only. If you want to amend, you can amend, make a board, go to a board, ethically get it approved, and resubmit for review. In a trial, you must use references which are from peer reviewed sources, as we discussed earlier, but they must follow a style, and preferably in Vancouver style, the references should be there. An important aspect, the intellectual property right. As we know that every intellectual property rights and values. The values must be studied in detail to safeguard the study against plagiarism and intellectual theft. In this big world, you never know who is going to steal your intellect or your work and presenting on his or her name. So always try to study these values. And because it is illegal to use others' IPR in your study without taking a prior permission, it must be clarified in the beginning that who would all be the stakeholders of IPR generated. If a good IPR is generated, everyone wants to encash that. But if it is clarified in the beginning that these persons, one, two, three, four persons, or n number of persons would be the stakeholder of these intellectual property rights, there won't be any conflict. This is called the conflict of interest. You must have seen in peer review publications that this study does not have any conflict of interest because the IPR values were defined in the beginning and no third party or no third person has any objection to those values. But if these values are valid and legit, we may go for patenting the outcomes and the process. Usually, processes are patented easily. At the end of the protocol trial, we must annex the case taking performer, the logistics required, the patient information sheet, the consent form, or any other document used in the study. Basically, this section contains all the performers, all the logistics, all the sheets which you are referring in your research, and that must be approved by a committee. For example, the case taking performer is approved, the validated score used in your study is approved, the consent form used in your study is approved, any other document, maybe a memorandum or maybe a legal document which is connected to your study, is approved from a bench of experts. Now, these are some of the quick bites. This is the clinical trial broadly has these headings which we have discussed. So I'm going to read in order that a trial should have a study summary, a study team, a list of abbreviations, followed by an introduction, which is a background information, the preclinical studies, the literature review consisting of prior research work, need of the study, what is the proposed or the expected role of that study, what is the study design phase of the study, the primary and the secondary endpoints, the population selection, the inclusion, exclusion, and sample size, how and when the screening and recruitment would be done, what would be the source of population, what is the screening process, what is the recruitment on or enrollment rules, where the study would be conducted, how it would be randomized with a flowchart, on what measures it would be studied, that primary outcome measure or the secondary outcome measure, what are the sensitivity, specificity of these outcome measures, what is the repetition of investigation, how frequently the investigation would be done in your trial, what is the follow-up frequency, how and when the treatment would be given to the patient, what are the study drugs, is it an individual study or the specific study or the constitutional drugs would be used, what is the treatment regimen, how and where the medicine would be procured and administered to the patient, what is the selection criteria, the potency and the dose selection, the prescription, the change in prescription, what steps we would take to maximize adherence and retention of participants in the study, how and when to withdraw the participants, what is the early withdrawal, what to do with the data of these withdrawn patients, what is our statistical plan, the sample size, the methods, the population for analysis, what are the safety or adverse events, the definition, the recording, the reporting, the unblinding procedures, the stopping rules, how and when the study would be monitored, the data handling, record keeping, source document, record retention, auditing, ethics, and DSMB, that is Data Safety Monitoring Board, who will finance the study, what all finances we need, what is our budget, what is the total grant and aid required for the study, which is breakdown up into headwise. What is the publication plan? What is the timeline? How and when the protocol would be amended with valid references, with IPR values, and with annexures we used in the clinical trial protocol. That is case taking, the logistic, the patient information sheet, the consent form, family history, or memorandum of understanding, whatever it is. These are some of the tips I want to give you, that we should avoid the term subject. Rather, we should use the patient or the participants because it seems more professional, ethical, and respectful. We should try not to deviate from clinical trial protocol as it is an ethical document which is developed with hardship, with hard work, keeping in mind various issues. However, there may be some situations when you have to deviate, but that deviation must be in consultation with the statistician. These three things always remember in mind: the outcome measure, endpoint, and objective defines your research. All other things support these three things, which are most important things in a research. You must mention your study team In the second page of the proposal, with all these headings, what is the degree, the address, the contact IDs, what is the role in the study, and whether that author has any experience in doing the similar work. You may download this post webinar or you may access this webinar for education purposes, and you can get in touch with me at my ID, that is info at the rate researchinhomeopathy.org. Just simple ID. And some of the useful links I've put on my website under this resource material section, which contains all the guidelines, the checklists, and the things needed to write a clinical trial protocol. You can go and check on my website. And I thank you all for your patient listening. I'm Dr. Saurav. I run a research website that is Initiative to Promote Research in Homeopathy. And my email ID is info at the rate researchinhomeopathy.org. You can visit my website, that is sauravarora.com, or you can go to researchinhomeopathy.org. And on my website, you can subscribe to free research updates like this webinar. You can follow me on Facebook and Twitter also with the username Homeo Research. That is facebook.com/Homeo Research or twitter.com/Homeo Research. And now I would be live on my website for half an hour for a live chat. If you have any question regarding this presentation, this webinar, clinical trial preparation or protocol, or any research related query, you may get in touch with me. I'm online at my website. I thank you all for your patient listening, and thank you. And you may show your support by visiting my website and go to the support section. Thank you.